Hello, for anyone who missed it or who needs a recap, we're going through 2.8 today, standard level cell respiration. So the first thing is to think about where this is taking place. So this is a picture of an animal cell here, so a eukaryotic cell, and we're focusing particularly on the mitochondria here and the cytoplasm, this region out here that's kind of watery, it's got some ions and enzymes and things in it as well. So this is where our reactions are going to be taking place. Now, respiration, that's one of many biochemical reactions that happens in living things, and we know that metabolism refers to all sort of our biochemistry, if you like, anything that sort of happens inside a cell. And we can talk about things that build larger molecules. They're anabolic reactions, so things like photosynthesis builds organic molecules from inorganic ones, and catabolic reactions break them down. So respiration is definitely a catabolic reaction. And a quick memory aid there, the anabolic steroids, if you've heard of them in the media before, they're the ones people often abuse to build muscle mass because they're building ones. So respiration, as we said, we're talking about the mitochondria here, and also a step that occurs in the cytoplasm. And the overall process of respiration is about breaking down organic molecules. In particular, we're looking at sugars, and the idea is to be able to extract the energy from those molecules to be able to make this stuff down here, this ATP. It's because ATP is the energy used by your cells for things like DNA replication, to move muscle filaments, to send electrical messengers, anything pretty much with life that requires an energy source. The energy your cells use is ATP. Problem is you can't eat ATP, you can't get it through your digestive system and around your bloodstream, but you can move sugars and amino acids and things around your bloodstream and get those into your cells. So all of your cells need a way of making ATP. And what is it? ATP is this molecule over here. It's short for adenosine triphosphate. So as you can see here, the tribe have got 3 phosphate groups. It's otherwise chemically, this part of it here is the same as this nucleotide over here in your DNA. So you'll see there's a lot of recycling in biology, which is some might use as an argument for evolution, that there's not just clearly defined different molecules with different purposes. That sometimes over the course of evolution, a chemical with a slight change has been fit for a particular purpose, and so it's been used in that way. So we've got here this ribose part and adenine, and then in DNA you would just have 1 phosphate because you have that phosphate sugar backbone here. But of course ATP we have 3, and it's this 3rd phosphate group over here with this really energy rich bond to the other 2 that can be reversibly attached and detached as a way of transferring energy around the cell. So we've got this high energy sort of phosphate bond here. We can think of then ATP as being a bit like a rechargeable battery. So this is our ATP over here. It's like our full battery. So this phosphate, when it comes off, it's not attached to anything with carbon anymore, so it's inorganic when it comes off. That breaks away and will connect and bond to other structures in the cell. So there'll be a transfer of energy in that process. So ATP is like your battery that gives energy to things that are happening in the cell. Once that phosphate comes off, as you might have guessed, if this one's ATP for triphosphate, this one's ADP for diphosphate. This is like the rundown battery, we've run out of energy, the low energy version. And your cells just cycle between the 2 of them. So they use ATP to do things, and then they charge it back up, the ADP, combine it with inorganic phosphate to make more ATP using cell respiration, using products of your digestion like sugar, breaking that down to make up a supply of ATP. So they're always cycling between the 2 forms in your cells. So that's the point. Cell respiration is about making sure your cells have lots of ATP ready to go to do stuff, so they have energy. Now, the first stage then in cell respiration is called glycolysis. You might be familiar seeing like lysosomes or other situations with the word lysis, which means splitting, and then our glyco for sugars over here. Remember glycogen. So glycolysis, that's our first stage. And look, you can actually modify other molecules. If you're on a low carb diet or you're starving, your body will use other energy sources like fats, and even if your body's wasting away, you'll break down your muscles and use your proteins as an energy source. So there are different biochemical pathways to get to this stage, but we're just going to look at glucose, which is our most common sugar form. So glycolysis then, this is just a very stylized kind of way of modelling our sugar, glucose over here. You'll remember that actually it's a hexagon with some other elements in it. Well, this is me in bad stylist work over here. But of course, we've got oxygen and hydrogen in here as well. But for all intents and purposes, just doing this kind of model here using the carbons, because what we're focusing on, to show you that our sugar starts off as a 6-carbon molecule. So what do we do in glycolysis? We split sugar. We break it in half, and that's what we get. And in the process, we can see our inorganic phosphate joined. We had our ADP over here, plus we can put a little I down the bottom to show that's inorganic phosphate, and we made ATP. Now that's actually happened twice. It's happened here and here. So for each half of the sugar, if you like, we've cut our sugar molecule into 2. So we now have 2 3-carbon molecules over here, and we have made 2 ATP. Now the name of this one is pyruvate or pyruvic acid. So if I get rid of some of these things, or look, now I've got all of these in the way. That's the problem with my stylus scribbles. If I just remove some so it's not quite so messy for you. That's not as easy as I thought it was going to be. At the end of the day, the big thing here, that it's in the cytoplasm. So we haven't actually gone into the mitochondria yet. It's actually more complex than just a single step, but for standard level, that's all you need to know. But all the steps are facilitated, if you like, or controlled or regulated by enzymes that are present in the cytoplasm. What you get those 2 molecules of pyruvate, these ones here, 2 of them, and 2 molecules of ATP, so charged up 2 batteries, if you like. 2 of our ATP have been made in glycolysis. So we've been able to break our sugar a little bit, and we've been able to get a little bit of energy out of it that we can use for the cell. So pause the video if you like, see if you can answer these. Quickly check. All right, the answers. First step, glycolysis, the products, pyruvate and ATP. How much ATP? 2. And where does it happen? In the cytoplasm. So where does it go next? So this funny little egg over here is my bad drawing of a mitochondrion. Now, if we have oxygen available, then the pyruvate can move into the mitochondrion and be completely broken down, and we get lots more ATP, another sort of 34 to 36 ATP, which is a lot more than the 2 we got in glycolysis. If we don't have oxygen, so that would be anaerobic conditions, then we can't get any more ATP. So we just have to do some rearranging of those molecules to recycle a few things needed in the cell. We can't have a bank up of pyruvate. So in some organisms that will be fermented into alcohol, ethanol and carbon dioxide. So ethanol is a 2-carbon molecule and carbon dioxide is 1. Or in us we get lactate or lactic acid. That's that, oxygen jet in your muscles. So there are pathways we can take. Obviously, most of the time, most organisms, this is the preference, to be able to really extract lots of energy from your food that you've gone to the trouble of hunting or chewing or whatever, digesting. You want to be able to get maximum yield of energy. But the key difference there, be really clear, anaerobic respiration, there is no further gain of ATP after glycolysis. So let's look at the aerobic though in a bit more detail. So we've said that's where oxygen is present. Our pyruvate, that's our 3 carbon molecule. So we've got 2 of those from our sugar. There's something called a link reaction. So in high level, we look into a little bit more detail about this. So this is the next stage, and then it heads into the mitochondrion. Now the mitochondrion, this kind of fluid part, it's like kind of the cytoplasm equivalent of the mitochondrion. We call that fluid part the matrix. And what we then turn the pyruvate into is acetyl-CoA, which is a 2 carbon molecule. So we're starting to break it down a bit more. And then the next stage, there's a whole cycle, a whole series of reactions. It gets back to the same starting point, so it can keep going again and again. And that's called the Krebs cycle. It's named after someone, Krebs. And in high level, we look at the stages a bit more and the electron carriers. In standard level, you just need to know that that's what it's called. And that is what breaks your carbon down all into carbon dioxide and water. And you get a little bit more ATP from this one. And then what you've done through that process, you've actually kind of charged up some other little intermediate molecules. And there's a whole lot of electrons involved that kind of have to go through a little turnstile or like a mill. We harvest all this energy on this membrane. These membranes are called cristae, these infoldings here inside our mitochondrion. And again, high level look at this in more detail, but that's the part, that's the critical part that makes heaps and heaps of ATP. So the idea is to get our pyruvate into our mitochondrion, have a link reaction, go through a cycle of reactions called the Krebs cycle, and then extract as much energy as we can from our molecules and break them down completely into that carbon dioxide and water. Now, I don't think, I might not have put it on this one because in the photosynthesis one, but the summary equation, we take C6H12O6, if that's the formula for our glucose, and we break that down to 6 carbon dioxide, that's to balance the carbon, and water here, 6 H2O, so 12 hydrogen atoms on either side. We have on this side of the equation, on the products on the right, we have got here 6 lots of 2, 12, and another 6. We've got 18 atoms of oxygen, and over here we've only got 6. So we need to balance this. Oxygen molecules in the air go around in pairs, O2. So that's an extra 12 atoms altogether of oxygen. Don't worry if you're not a chemist and you're not interested in balancing equations. What I'm trying to say here though is that in order to break down our sugar, we need oxygen. That's why this is aerobic, oxygen requiring, because we actually need the oxygen to pair up with our carbon and hydrogen to make it all into carbon dioxide and water. Otherwise, we can't completely break it down, and we have to go the anaerobic route. And this is why we need oxygen. When people ask you why you have to breathe, why do you even need it? Why do your cells need oxygen? Well, they need oxygen because you need to break down your food, because if you don't break down your food, you don't have ATP. So if your cells don't have ATP, they die. That's why you need oxygen. So the alternative then, which we can't survive off, you can have short bursts of a bit of anaerobic respiration in people, but not a lot. Whereas some other organisms like yeast and some bacteria can survive anaerobically. So this is our no oxygen environment where anaerobic respiration will occur. So the pyruvate does not go into the mitochondrion, so it just stays in the cytoplasm. And we've got no more ATP. So don't forget though, we've got the 2 from glycolysis. So you've got a tiny bit of energy out of our sugar. In us, we get lactate. But one of the things you see in the food production industry, it's really important that yeast make is carbon dioxide and ethanol. So ethanol being our alcohol that's in beer and wine spirits, and the carbon dioxide bubbles when you leave your dough to sit and rise, and when you get those little carbon dioxide bubbles in your dough, and then you bake it and they expand even more, and then the yeast actually is killed off as you cook it. So we have that's really important in industry. And the reason I put this picture here, this poor guy that I'll try not to make too much fun of him down here, but I've just put it there because people often use the expression beer gut. And look, you can have an enlarged middle area if you've got a bit of liver damage from all that alcohol. But the point I'm trying to make is actually just that alcohol has tons of calories in it because it's barely modified sugar, really. If you only get 2 ATP out of every sugar molecule in this anaerobic pathway, that means there's a ton of energy still in your ethanol. So gram for gram, alcohol is about the same as sugar or carbs in terms of its energy content. So consuming a lot of it is not good for your health on a lot of levels. Now, when we talk about the aerobic part, look, some books have slightly different numbers because it's a pretty hard thing to quantify, and things enter the Krebs cycle at sort of different stages. But generally in the aerobic part, we've got that glycolysis. So this is common to aerobic and anaerobic. Both of these occur in a cytoplasm, glycolysis, and then aerobic stops. And our pyruvic acid or pyruvate has to be turned into lactate or ethanol with no more ATP. If we have oxygen, then we can go into our mitochondrium. And there's actually, it's not on this picture, but there's a little link reaction, the first stage of losing a carbon, and then the Krebs cycle breaks it all up. And what we call this electron transport system or electron transport chain is our big mother load of ATP. And only the names of these processes are needed at standard level, and identifying that this is the part that needs oxygen and our output is carbon dioxide and water. And all this ATP coming from this aerobic system in the mitochondrium. So our summary, if this hopefully this helps you, there's our glucose, again, glycolysis, 2 ATP, pyruvate. It could be fermented, we could get lactate in animal cells or ethanol and a bit of carbon dioxide in yeast cells. Or if we've got oxygen, we have a link reaction in the mitochondrion, we make something called acetyl-CoA and some carbon dioxide. We undergo the Krebs cycle and the electron transport chain to break it up completely and extract lots of ATP. So we've got this kind of 34, sometimes up to 36 extra ATP in this stage. So it's a big difference in the energy yield. Now in the lab, and thinking about IAs, we can measure respiration. A direct measure is talking about what things are in the chemical equation. So if you look at how much oxygen is consumed, or how much carbon dioxide is produced, that is a direct measure of the rate of respiration. So you can do things with like pH probes because carbon dioxide makes an acid. You can do things with dissolved oxygen probes as well that measure the oxygen in the air in a test tube. Or you can do things indirectly with temperature. Direct things are generally better, but doesn't mean you can't do it or it's not an alternative to look at a temperature change, because a byproduct of respiration is heat, which is why we say your metabolic rate increases when you're cold. You burn more fuel to keep you warm. And then thinking about things that affect the rate of respiration, you could look at age of germination, temperature, or conditions that they're in, comparing species or ripeness. There are lots of things that you could look at. And look, that's up to you. I'm going to leave it at that because this is meant to be short and sweet where possible. And you've got lots of resources based around IA ideas that you can look at. All right, let me know if you have any questions. And hello, shout out to anybody who hasn't been able to make it to class.